we have to start another topic, that is enzymes. Enzymes, you have already studied the catalyst in your inorganic chemistry. Catalyst में क्या पढ़ा था आपने? That increase the rate of reaction by decreasing the energy of activation. So catalysts are, that is the substances, inorganic substances, which increase the rate of reaction by decreasing the energy of activation. तो इसी तरह एंजाइम जो है इन बायोकेमिस्ट्री particularly they are present in animal as well as in vegetable. They are produced in animal as well as in vegetables, and they are known as biocatalysts. They are known as biocatalyst. तो बायोकैटलिस्ट जो हैं ये क्या किस किस्म की चीज़ें होती हैं? दे आर एक्चुअली दे आर मोस्टली प्रोटीन इन नेचर. They are mostly protein in nature, and they are required in small amount. They are required in small amount. They increase the rate of reaction by decreasing the energy of activation. वही the same. They increase the rate of reaction by decreasing the energy of activation. And furthermore, they do not initiate the reaction. रिएक्शन को इनिशिएट नहीं करते, इनिशिएट नहीं बल्कि कैटलाइज करते हैं. दे इंक्रीज द रेट ऑफ रिएक्शन, इनिशिएट नहीं करते. इन द एब्सेंस ऑफ एंजाइम द रिएक्शन बिकम वेरी स्लो, एक्सट्रीमली स्लो. होता ज़रूर है बट इट इज़ एक्सट्रीमली स्लो. द सब्सटेंस ऑन विच द एंजाइम एक्ट उसे कहते हैं हम सबस्ट्रेट. जिससे पे अमल करते हैं एंजाइम, the substance on which they act, they are known as what? Substrate. They show activity in vitro and as well as in vivo. They show activity in vitro and in vivo. In vitro mean that in a test tube. That's a common मामला साड़ा सारा. They show activity in vitro, that mean in a test tube or in a vessel, beaker or in a big vessel, fermenter, as well as in animal or in vegetable, जहाँ से produce होते हैं. That's mean in vivo. तो इस किस्म में they are so of course they are effective to the temperature as well as to the pH. pH से और टेम्परेचर से ये इफ़ेक्ट होते हैं. ख़ास टेम्परेचर पे इनकी एक्टिविटी ज़्यादा होती है, जैसे कहते हैं ऑप्टिमम टेम्परेचर. एक ख़ास पीएच पे उन पे ख़ास पीएच पे इनकी एक्टिविटी ज़्यादा होती है, उसे हम कहते हैं ऑप्टिमम पीएच यानी हो. तो ये एंजाइम इस किस्म की चीज़ होती है. अब ये इसका मैकेनिज़्म क्या है? व्हाट इज़ द मैकेनिज़्म ऑफ एंजाइम? एंजाइम रिएक्शन का मैकेनिज़्म इन्वॉल्व 3 steps. इन द फर्स्ट स्टेप the enzyme combined with the substrate. Mechanism. Of enzyme. Mechanism of enzyme. It involves 3 steps. In the first step, the enzyme combined with the substrate to form enzyme-substrate complex. In the first step, the enzyme combined with the substance with the substrate to form enzyme substrate complex, ES. इसमें ज़रा आप सोचें थोड़ा सा कि किस किस्म की बॉन्डिंग होगी बिटवीन एंजाइम एंड सबस्ट्रेट. कोवलेंट बॉन्डिंग नहीं होगी. हाइड्रोजन बॉन्डिंग इज़ पॉसिबल, आयनिक बॉन्डिंग इज़ पॉसिबल, वैन डर वाल्स फोर्सेस आर पॉसिबल. वीक बॉन्डिंग होगी, वैन डर वाल्स फोर्सेस, आयनिक बॉन्डिंग, हाइड्रोजन बॉन्डिंग, इस किस्म की बॉन्डिंग होगी. और एंजाइम पे ख़ास एक्टिव साइट होती है. एंजाइम पे ख़ास एक्टिव साइट होती है. मतलब एंजाइम का स्ट्रक्चर जो है आपने टर्शियरी स्ट्रक्चर में पढ़ा हुआ होगा, वो ऐसे होता है इस तरह. NH2 एंजाइम का स्ट्रक्चर इस किस्म का होता है, तो इसमें जो एक्टिव साइट होती हैं वो ऐसे होती हैं, ऐसे जहाँ ये क्रॉस करते हैं। एक्टिव साइट actually attract the substrate toward itself, and it combined to the active site and product is formed. Just combine with the substrate, with the active site of the enzyme, and product is formed. Enzyme and the substrate, they are complementary to one another. Complementary. Complementary to one another. It is just like a lock and key relationship. Lock and key relationship. Lock is that is substrate, and key is that is the enzyme. Lock and key in relationship. And of course, in other words, we say it is the induced fit mechanism or induced fit arrangement. Induced fit arrangement or mechanism between the enzyme and the substrate to form the enzymic reaction. Induced fit arrangement or mechanism. Arrangement between the enzyme and the substrate to form enzymic reaction. तो इस किस्म का E और S के दरमियान इस किस्म का रिलेशनशिप होता है. In the second stage, the enzyme substrate complex changes into enzyme product complex, EP. In the second stage, enzyme substrate complex changes into enzyme product complex, and in third stage, enzyme product complex, enzyme plus product. The enzyme released, and release enzyme again that combine with the substance to form enzyme complex, substrate complex. बाज़ दफ़ा हम कहते हैं एंजाइम हिस्सा ही नहीं लेता है. Sometimes we say the enzyme do not take part in reaction क्योंकि रिलीज होता है. लेकिन कुछ थोड़ा बहुत फ़र्क पे आ जाता है जो हम नहीं मालूम कर सकते. We cannot determine it by ordinary methods. फ़र्क पैंदा है. अगर ज़्यादा टाइम में इस्तेमाल हो ना एंजाइम तो एंजाइम एक्टिविटी ख़त्म ही हो जाती है. बार बार उसे इस्तेमाल करें तो एंजाइम एक्टिविटी ख़त्म भी हो जाती है. कहते हम यही हैं, वो यार ऐसा नहीं लेता लेकिन होता है. It takes part in the reaction. Anyhow, ये सारी रिसर्च की बातें हैं. हाँ, ओके. नाउ अब ये है कि एंजाइम होता क्या है, व्हाट इट इज़, केमिकली व्हाट इट इज़, केमिकल नेचर ऑफ एंजाइम, केमिकल नेचर ऑफ एंजाइम. अच्छा, एक बात रहेगी वैसे. वी हैव नॉट डिस्क्राइब the example for the enzyme that we say that the enzyme catalyzes the reaction by decreasing the energy of activation. उसके लिए एक एग्जाम्पल आपने पढ़नी है. वो एग्जाम्पल that is the decomposition of hydrogen peroxide. Decomposition of hydrogen peroxide. Hydrogen peroxide at normal temperature, it is decompose itself to water and oxygen. But the energy required to decompose 1 mole of hydrogen peroxide, that is 18,000 calories per mole. 1 मोल हाइड्रोजन परऑक्साइड को 1 मोल हाइड्रोजन परऑक्साइड को कन्वर्ट करने के लिए एनर्जी जो चाहिए दैट इज 18,000 कैलोरीज normal temperature, but in the presence of a catalyst, catalyst क्या है? Platinum. The same reaction in the presence of catalyst, platinum, the same product, but the energy which is required in the presence of catalyst, that is 11,000 calories. The energy which is required, that is 11,000 calories. Catalyst ने काम किया, energy required to decompose 1 mole of hydrogen peroxide in the presence of catalyst, that is platinum, डिक्रीज हो गई. हम कहते हैं ना, केमिस्ट्री में भी यही कहते हैं ना कि एंजाइम जो है वो अपना रेट ऑफ रिएक्शन को इंक्रीज करता है बाय डिक्रीजिंग द एनर्जी ऑफ एक्टिवेशन. तो दिस इज़ दी, बट इन केस ऑफ एंजाइम same reaction takes place in the presence of enzyme. एंजाइम कौन सा है? उसे कैटालेज कहते हैं. The enzyme is required, that is catalase, to decompose hydrogen peroxide into water, and the energy is required 2,000 calories. The energy required, that is 2,000 calories, to decompose 1 mole of hydrogen peroxide into water and oxygen, and the energy required, that is 2,000 calories only. 2,000 calories only. You just compare कि कितनी एनर्जी कम होती है जिस पे कि ये रिएक्शन हो जाता है इन द प्रेजेंस ऑफ एंजाइम. It has been observed. This is the function of enzyme. It catalyzes the reaction. That means it increases the rate of reaction by decreasing the energy of activation. हाँ जी. Catalase is the enzyme. Platinum is catalyst. So this is the example, and that has been observed that the enzyme that can, of course, change the substrate into product. How much molecules? 100 to 1,000 molecules per second. This much number of molecules, they can be converted by 1 molecule of enzyme per second. This much number of molecule of the substrate can be converted to the product. This much number of molecules of the substrate into product by 1 molecule of enzyme per second. ये एग्जाम्पल है. नेक्स्ट कम एंजाइम एक्टिविटी. नहीं, केमिकल नेचर ऑफ एंजाइम. Chemical nature of enzyme. Enzyme actually consists of 2 parts. 2 हिस्से होते हैं उसके. It consists of 2 parts. One is pure enzyme, pure protein rather. Pure protein. Another part of it, that is the factors. Factors mean the substances which facilitate the enzyme to catalyze the reaction. Factor means what? The substances which facilitate the enzymic reaction. Factors दो किस्म के, two types of factors. One is organic, the other is inorganic. Organic and inorganic. ये जो है, that is one part of it is pure protein, जिसको हम कहते हैं apo enzyme. One factor of chemical nature of enzyme, that's pure protein, which is known as apo enzyme. The other is, that is factors. Factors दो किस्म के हैं, organic and inorganic. Organic are known as coenzyme. Coenzyme. And inorganic, they are inorganic ions. They are known as cofactor. Inorganic ions, which are known as what? Cofactors. Chemical nature of enzyme. One is pure protein, which is known as apoenzyme. The others are factors, organic and inorganic. Organic is known as coenzyme, and inorganic, that's known as cofactors. The pure protein and all the factors, they are known as what? Holoenzyme. The pure protein along with factors, your apoenzyme along with factors, they are known as holoenzyme. They are known as holoenzyme. Now about the cofactors, inorganic ones. For example, in case of urease is an enzyme which decomposes the urea into carbon dioxide and water. Urease is an enzyme which converts the urea into carbon dioxide and water. Usme nickel is as what? That is the cofactor. Nickel ion. Carbonic anhydrase. This is an enzyme which of course decompose कौन सा एसिड है? कार्बनिक एसिड into carbon dioxide water. तो इसके लिए जो एंजाइम है, that's known as carbonic anhydrase. It contains zinc. Carbonic anhydrase, which catalyzes this reaction. Carbonic acid changes into carbon dioxide, and carbon dioxide plus water changes into this one. Reversible है. Zinc. Well, let's go in case of pyruvic dehydrogenase. Pyruvic dehydrogenase. इसमें है मैग्नीशियम. magnesium is as what cofactors. तो इस तरह the cofactors are there. Now come the coenzyme. Coenzyme, they are organic molecules, organic substances such as NAD, nicotine adenine dinucleotide, oxidized form, NADH2, nicotine adenine dinucleotide in the reduced form, FAD, flavor adenine dinucleotide in the oxidized form, and flavor adenine dinucleotide H2 reduced form of it. इसके अलावा lot of NADPH2, GTP, TTP, CTP, DDP, and so on and so forth. They're all what coenzyme. They're all what coenzyme. ATP वगैरह वगैरह. अब इनका जरा स्ट्रक्चर देख लें. In case of NAD, nicotinamide adenine dinucleotide, it is like this. And that's here. Nicotinamide along with the ribose two phosphoric acid, ऐसे स्ट्रक्चर होता है इसका. नहीं, ये गड़बड़ हो गया. अच्छा, CH2 here. स्ट्रक्चर खराब हो गया. So this is that the NAD and changes to NADH2. This is the reduced form of it. This is reduced form, this is oxidized form. Oxidized form and reduced form. रिड्यूस फॉर्म में रिएक्शन यहाँ होता है, यहाँ रिएक्शन होता है और दिस चेंजेस इनटू व्हाट, ऐसे। And that's those 1, 2, 3. 1, 2, 3. H. प्लस प्रोटॉन ठीक है। ऐसे और दिस इज द reduced form of it. 2 hydrogen atom is taken away, एक हाइड्रोजन इधर और एक हाइड्रोजन in the form of proton. In the form of proton. Return नहीं लाना, just see that wherever we use the SNAD, particularly in the metabolism, you must know कोई ऐसा molecule होता है जो which take away the hydrogen. ये reaction reversible भी हो सकता है, particularly it is involved in oxidation reduction reaction. ऐसे हो तो ये oxidation, ऐसे हो तो फिर it is reduction. It is NAD, nicotinamide adenine dinucleotide. Another one that is FAD, flavin adenine dinucleotide. उसमें भी क्योंकि एडेनिन इज इन्वॉल्व, तो एडेनिन में ये इस तरह है कि ये इंस्टेड ऑफ द राइबोज यू हैव व्हाट द रिबिटॉल, द अल्कोहल फॉर्म ऑफ इट, रिबिटॉल. ऐसे दिस इज ये पार्ट जो है दैट इज हम इसे कहते हैं FAD, फ्लेवन adenine dinucleotide, flavin adenine dinucleotide, flavin adenine dinucleotide. न्यूक्लियोटाइड में क्या क्या होता है? हाइड्रोजन बेस, शुगर एंड फास्फोरिक एसिड. Another phosphoric acid, this is ribitol, the alcohol form of the ribose, and this is of course the flavin, riboflavin जिसे कहते हैं. Actually it is from riboflavin. So it is the molecule of FAD and changes into FADH2, again causing what, the oxidation by taking away the hydrogen of the molecule. तो इस पार्ट में यहाँ here reaction takes place. When it changes into FADH2, then this double bond is ख़त्म हो जाता है, ये डबल बॉन्ड ख़त्म हो जाता है, ये गलत हो गया काम, नाइट्रोजन इतने होनी चाहिए सी। हाँ जी, हाँ ये एट द एंड आना, एट द एंड ऑफ द क्लास, एट द एंड ऑफ द क्लास। ये इसका स्ट्रक्चर यूँ है, ये CH2 कंबाइन विद दैट दिस नाइट्रोजन, यहाँ नहीं, इस नाइट्रोजन साथ में लिव। ओके, व्हेन द NaDH2 FADH2 इज फॉर्म, तो दिस डबल बॉन्ड इस ख़त्म हो जाता है, ये ख़त्म हो जाता है, और इसके साथ हाइड्रोजन लग जाती है, और इसके साथ हाइड्रोजन लग जाती है, और यहाँ एक डबल बॉन्ड क्रिएट हो जाता है। नाउ इट इज़ द फॉर्म ऑफ FADH2। FADH2 फार्मूले ठीक है, एक दफ़ा देख लो। विल यूज़ दैट FAD, NAD, NADH2, FADH2, तो आपके जहन में होना चाहिए ये है क्या, बस और कुछ नहीं, ज़ुबानी याद ना रखना। This is about the coenzyme, that is NAD and FAD. Now comes the enzyme activity. This is important. Enzyme activity. Try to understand it. There are three types of enzyme activity. One is known as international unit. International unit, IU. International unit. It is IU. International unit. It is the amount of enzyme required to transform 1 micromole of the substrate into product. It is what? It is the amount of enzyme which convert 1 micromole of the substrate into product. Which convert 1 micromole of the substrate into product is known as in 1 minute time भी है per minute at 25 degree centigrade temperature. क्या है international unit? It is the amount of enzyme required to convert 1 micromole of the substrate into product per minute at 25 degree centigrade temperature. International unit. Other is, that is cattle. Cattle is what? That is the amount of enzyme in terms of international unit required to convert 1 mole of the substrate into product per second. The amount of enzyme in terms of international unit required to convert 1 mole of the substrate. 1 mole of the substrate per second at 25 degree centigrade temperature. Cattle, The third is that is specific activity. Specific activity. Specific activity. This is the amount of enzyme in terms of international unit present per milligram of enzyme protein, pure protein, enzyme protein or pure protein. The amount of enzyme in terms of international unit present per milligram of enzyme protein or pure protein. So these are the three enzyme activities. कितना टाइम है? हो गया? चलो आहो CR कौन है? ये पकड़ो, खुद डिस्ट्रीब्यूट करो.